So first of all, thank you very much for giving me the opportunity to function as an advocate for this exciting new cancer-specific tumor marker of circulating tumor DNA. It will give a little bit of a different spin on probably the discussion, as this is not a tissue-based marker. I'm a medical oncologist by training, and I'm still doing that. And my major disease focuses on brain cancers. I have, unfortunately, nothing financially to disclose. And if you're wondering where my accent comes from, I'm from Germany, so that's to disclose the rest. I'm going to start with pointing out that I'm going to structure my brief presentation into 4 sections. First of all, I'm trying to illustrate what circulating tumor DNA is, and then how it can be detected at the present time in plasma of patients. I will focus on the potential use in clinical oncology, and last but not least on the clinical challenges in bringing this into a clinical setting. Over the past years, we have learned a lot about the genetic makeup of a variety of cancers. And this is based on the tremendous effort of multiple groups doing large genome-wide analysis of the cancer genome. So we have a fairly strong background that we can base any more targeted, genetically targeted diagnostic tests on. So there are a number of genetic alterations that a cancer can harbor. And this slide illustrates that essentially the majority of these are still single nucleotide substitutions or point mutations. So how do we get access to these cancer-specific genetic alterations? And there are essentially 2 main sources for these mutations and other alterations. One of them is tissue, and as Dr. Williams illustrated, it comes with certain challenges from a laboratory basis, as well as from a pathology basis, but also practically. Patients have to undergo a biopsy, have to have surgery, and the number of time points you can do this in a patient are limited. There's paraffin-embedded DNA that is often stored for long periods of time, can degrade. There's sampling issue. And we have frozen tissue that is a little bit more appealing. We can also look into blood and other bodily fluids. And if we look in those compartments, we can look at cell-free DNA, and we will also find circulating tumor cells. So what is circulating tumor DNA in a nutshell? So first of all, this is not synonymous with circulating tumor cells, which you find out quickly if you submit a grant on circulating tumor cells and it comes back and people think you're talking about tumor cells. Cell-free DNA is released during cellular turnover, at least that's what we believe at this point in time. The circulating tumor DNA fragments, which I'm going to call ctDNA fragments during this talk, are very small. They are 180 to 200 base pairs long. The interesting part is that these fragments, although they are small and they are sort of random, depending on what is floating in the blood, they can contain tumor-specific somatic mutations. And that's exactly what the concept of circulating tumor DNA is going after. So this is a schema from a actually excellent review article on circulating nucleic acids that illustrates the origin of circulating tumor DNA and also what we eventually find in the bloodstream. On the left side of the slide, you see in blue the cancer cells that are undergoing apoptosis or necrosis and that shed cell-free DNA into the blood. On the right side, you also see in green normal cells that can also die, that can also shed DNA into the bloodstream. You find in the blood, if you just took a blood sample, you can certainly find mutated tumor DNA, you can find normal DNA, you can find whole cells, you can find rearrangements, all kinds of things. So when we take a snapshot view of circulating tumor DNA in plasma, we find a lot of normal DNA. normal DNA mainly from tissues that are undergoing, as normal cells, a high cellular turnover. These are cells in the bone marrow, these are cells in the GI tract, cells in the skin, and certainly other tissues too. In the pool of cell-free DNA in the blood, in cancer patients, we also find a small fraction, sometimes smaller, sometimes larger, depending on the tumor type and stage of cell-free tumor-related DNA. As you can imagine, if a patient undergoes a traumatic procedure, such as surgery, or has a trauma, or you give radiation or chemotherapy, the proportions of tumor DNA and normal DNA can change. Furthermore, there can be variations between individuals, from one person to the other. So what do we get if we take a blood sample from a patient with cancer that harbors some somatic mutations? So we spin it down. We have usually the majority of it will be plasma, which contains trace amounts of cell-free DNA, of which trace amounts might be from the cancer. And we have cellular components, and we have trace amounts of circulating tumor cells. So we are essentially faced with the proverbial problem of finding a needle in the haystack. So as an example, if you have 5 mutant tumor DNA fragments in a pool of 10,000 DNA fragments, that's a fairly small proportion. If we used a conventional semi-quantitative PCR with a sensitivity or with a detection threshold of 1%, we might not detect the tumor DNA and we would not be able to quantify it. So one way to address this in a fairly simple but also very complicated and way is the concept of so-called digital genomics that Bert Vogelstein and Ken Kinsler started to work on in the late 1990s. What this essentially means, first of all, digital does not mean electronics, doesn't mean iPhone. It means, digital means essentially you look at every single DNA fragment that is floating in the blood and you determine with the test, is this wild type or mutant? Might not be mutant, 0 or 1, 0, 1, like in a computer. And the way to think about this is you have 112 George Washingtons on this picture, and you try to find out which of this. That's right. So which of the ones might be slightly different? If you had a test that had a 1% detection level, well, you might not see it, and you might just look the same as the wild type. However, if you looked at every single of these George Washingtons individually, eventually you would find the one that is a little bit different. And that's the fundamental basis of digital genomics, digital PCR. So we have a lot of diagnostic methods, a lot of toys we can use to try to be as good as possible in identifying tumor-related DNA in plasma. So on the left side of this scale, you see the detection limit of more conventional PCR-based assays. We're going down to digital PCR-based assays with a detection level of about 1 in 10,000 or better, including so-called beaming, which I will explain in more detail, droplet PCR-based assays. And then we can also look at several mutations at a time. We can do direct sequencing from plasma with new generation sequencing approaches. So one example I wanted to give, which was one of the earlier methods that had been published, is the so-called beaming. Beaming stands for Beads, Emulsion, PCR, Amplification, and Magnetics. And I'm going to walk you through this busy slide from left to right. So on the left side, you have the patient sample. And after isolation of the DNA, you have a random mix of wild-type DNA and trace amounts of tumor DNA mixed in. The first step is an amplification. The second step is essentially a way to separate all of the different DNA fragments into single PCR compartments. And with beaming, it's done within water and oil emulsions. We have billions of bubbles that have, statistically spoken, one or none of the DNA fragments, so that you can really have individual PCR reactions for either wild type or mutant. And in this bubble is also a magnetic bead. The magnetic bead beats in a sequence-specific way to the DNA fragment or to the target gene region. And so these beads will be loaded with that specific gene locus, and you lyse the bubbles, and in the end, with allele-specific hybridization, with little probes that bind specifically to the either mutated or wild-type point in this DNA fragment that was amplified, which is also fluorescent. You can then count every individual event. And this way, with flow cytometry, you can determine not only if a patient has a mutation, but you can also quantify it, and you can literally say, give a certain percentage of mutated DNA at a certain period of time. It's a fairly robust assay, and the detection rate is, and this is a conservative way to put this, about 1 in 10,000. So liquid biopsy is the term that we now use to describe how we can use circulating biomarkers. And cell-free DNA or circulating tumor DNA is one example. And a liquid biopsy is essentially, I'm going to use in the next slide as a term, it has a couple of appealing features. One of them, it's not invasive. It's a blood draw. You can take multiple specimens. A patient undergoing standard chemotherapy or is on an investigational protocol can have a number of blood draws that can be tacked onto standard blood draws, so it's not a real burden for patients. It's a highly specific, highly sensitive assay that we have. Actually, we have multiple highly specific assays that exist for these, for detection of circulating tumor DNA. We have a fresh source of DNA. Every time we get the blood, it's fresh DNA. And we have no sampling bias. By saying no sampling bias, I mean in a patient with a certain heterogeneity of genetic changes within the tumor, for example, widespread metastatic disease, if we get a blood sample, we would, if all mutations are represented in the blood, we would be able to detect that mutation. We are not bound to a biopsy that has to hit that specific disease-specific site that harbors a specific mutation. So what are the potential applications in oncology? And it's kind of fun to think about what we could do with this because it's such a versatile tool. So we could non-invasively determine the mutation status of a tumor. We can monitor disease burden. We can track molecular development of molecular resistance in patients in a non-invasive way over time. And we could potentially, if these assays are sensitive enough, detect minimally residual disease. For example, after potentially curative upfront therapy with surgery in low-grade colorectal cancer, breast cancer, and others. And if the assay, again, if it was sensitive enough, it could be helpful for early detection. Circulating tumor DNA might not be the right assay for every cancer. In a larger screening project looking at a number of different solid tumors, we found that in a number of cancers, we can detect at advanced stage, we can detect circulating tumor DNA in virtually all patients. However, there are other cancers where we cannot. And if you work in the field of breast cancer, lung cancer, and so forth, it's good news. If you work like me in the field of brain cancer, not so good. If you look to the very right end of the spectrum, you see that in gliomas, we can only in about 1 in 10 patients, even with sophisticated methods, detect circulating tumor DNA. Also, tumor stage seems to be important, which appears intuitively obvious that with higher stage and more tumor, we have higher rates of detection. So how do these studies look like? This is the most simple, straightforward example of how this can be done. With standard traditional sequencing, mutation can be detected in tissue, and then we will try to see if with with a digital PCR-based approach, can we find this mutation in plasma? And this has been illustrated with a couple of different techniques, and that seems to work. Once we know a patient's mutation, or if you look at tumor burden, it doesn't matter which mutation it is. Is it a driver mutation? Is it not? We don't care. As long as we have one genetic alteration that we can detect reliably that is important for the patient's cancer and that represents the patient's cancer burden, then we can use it as an individual cancer biomarker. So this is a slide illustrating dynamic tumor changes over time in a patient who undergoes surgery and chemotherapy, multiple lines of treatment for colorectal cancer. So you could also take a snapshot. You could take a snapshot to see what mutations are present in a certain patient. For example, this is of importance in colorectal cancer when we think about EGFR targeted therapy. The presence of a KRAS mutation is associated with less of a response. And so if we could bypass a traditional biopsy or looking at tissue and could get a plasma sample, that might be a little bit more elegant. And we can see in both breast cancer and colon cancer that the concordance is actually quite good. And furthermore, because this is a dynamic marker, we have samples over time, we can look at multiple mutations. We can trace and detect the emergence of mutations that may or may not in the future guide changes in therapy. So the clinical implementation of circulating tumor DNA is a little bit challenging, as is every other molecular test. So to date, there have not been any clinical trials that prospectively validated circulating tumor DNA as a cancer marker. We have a couple of hurdles. We certainly have practical challenges, and I think Dr. Williams has highlighted this, that we cannot pay enough attention on making sure we have the same process for every sample if we really want to have a cohort where we want to draw comparisons between samples and patients. We need better ways to collect and store samples. High cost, these are expensive tests. They become cheaper over time. However, for every new study, certainly we'll probably use tests that are very expensive. So there needs to be good funding. There are administrative barriers. And I put this as investigator time commitment. I mean, you have to find people who will have the time to devote a good portion of their professional life on really making this work. And then biological limitations. For example, looking at minimal residual disease, you might need very large numbers of patients to actually come to statistical significance. So where should we start? So it would be good, and this is a little bit of a brainstorming process, and it might actually continue throughout this workshop. It would be good to have dedicated funding and personal resources for large-scale companion studies to tack these circulating tumor DNA-based assays onto prospective clinical trials. We need better standardization of specimen sampling, processing, and storage. And we should start with straightforward questions, with fairly simple studies so that we can establish a methodology before making it more complicated. And we should probably involve experts in the field early on in clinical trial design. So in the year 2014, as a summary, what do we have? Circulating tumor DNA is a highly sensitive, specific, depending on the assay biomarker, and it is dynamic. So the difference to a lot of tissue-based markers, this is a dynamic marker where you can see changes over time. Most assays are both qualitative and quantitative. We can get a snapshot of the genetic makeup of the tumor and of the changes in tumor burden over time. We have many potential clinical applications. It's not the solution for cancer diagnosis for every type. And we have seen this, that different cancer types have different detectability and so forth. So where are we right now? What do we have? We have good or very good proof of principle data. And what we need is prospective validation, large-scale studies to determine the value of circulating tumor DNA for clinical practice. And I wanted to thank the Institute of Medicine for inviting me, our patients who volunteer for all these blood draws, and a lot of colleagues and friends who did a lot of this work. Thank you very much.
Thanks, Matthias. And next, also again, we'll be talking about other types of tests. So Neil Hayes from University of Carolina, Chapel Hill will talk about RNA-seq testing.